Good afternoon. I'm Will Meurer, and I'm one of the course directors for the NINDS Clinical Trials Methodology course. Thanks for coming. We're going to have a quick and action-packed webinar on asking a good early phase clinical trial question. So I want to acknowledge NINDS for funding our course, and both through the R25 and the Data Coordinating Center to the University of Iowa with Dr. Coffey, and also the American Academy of Neurology, which has been a gracious supporter of the course in many ways. Here are my disclosures. They're available on request, but there wasn't anything really nasty there. All right, so here are some quick objectives. Consider the purpose of clinical trials, consider the goals of clinical trials, and understand the potential output of clinical trials. The slides that I am presenting today have been derived from some various sources. Some slides were actually from a recent presentation about a new clinical trial called IceCap that will hopefully be funded by NHLBI that I gave at our department's research forum. There's a link there to that. There's some prior webinars, which you can find at the webinar tab at our website, neurotrials.training. A 2014 version of this where Bill Barson asked a good clinical trial question. And in fact, I actually used slides that he discarded. I used his sort of outtake slides, some of his outtake slides. His presentation's a little different. And then 2017, Roger Lewis gave an early phase trials goals and questions talk where I will briefly go through those. One thing that I would say is good to review. Last year we had Dr. Thabane, who gave a great webinar on pilot and feasibility studies. Since many of you may be designing projects that are in that space, it's something that I highly recommend checking out. So if you have questions, I will pause at each section header. Looks like this picture here. And if you type them in as comments, I'll bring back up the comment box and look for them there. I'll try to address others as I go along, depending on what Joy sends my way. So, okay, part one, scope of the problem for clinical trials. All right, so this is a stink bug. These, it's sort of still winter in Michigan, but these guys are going to be all over the place in not too long. So what are some of the reasons clinical trials stink? Well, this is an analogy that sometimes we don't think of clinical trials in the right way. But if we think of them as a diagnostic test, in medical school, many of us learned a lot about sensitivity and specificity and how well a diagnostic test performs. So if you think about this, if you're looking for an effective treatment, your type 2 error or 1 minus your type 2 error is the power or the true positive rate for a diagnostic test. Your type 1 error or the alpha is the significance level, that your false positive rate. So this is one way of thinking what you're trying to accomplish in a clinical trial. In the end, clinical trials are models. There's some form of a statistical or biological, philosophical, social science, epidemiological model where we need to make a ton of assumptions to run them. Some key assumptions are that the dose from animal models is close to what would work in humans. We often assume there's no heterogeneity of effect based on prognosis, gender, sex, age, that subgroups all respond equally, or that we exclude some subgroups. These patients are too bad off to be helped by our intervention, or they're too well off. They'll just do just fine no matter what. We can sometimes use an effect size to create a reasonable sample size. We like to do acute stroke studies that are like around 1,500 patients because that's what we do. But we probably would be willing to find new acute stroke treatments that had a smaller effect. It just would require many, many more patients. There can be noise in outcome measures, and how well that can be understood and overcome is not always clear early. And is the duration of treatment practical? Can you put somebody on an infusion for a month? It depends. But For some treatments, that may be what the biology tells you you need to do. So in the end, we do make a number of compromises to reduce the number of parameters and to make this solvable so that we're like, gosh, the patients need us to do a trial in this space. So yeah, there's lots of questions here, but we got to get something going on. And in the early trial space, it's hard because you have to know a lot about a lot of these things, but you also have to think about how many assumptions you're making and trying to inventory them. Not to say that you can address all of these questions in a small clinical trial, but you have to think about what those assumptions are and sort of inventory them and think about them. Going back to that analogy of we are looking at a model to find treatments that work, this is something called the therapeutic response surface, and the idea here is that there's some regimen, some treatment regimen, whether this is a combination of for a drug, a dosage schedule plus a dose, or a combination of drugs. There's some regimen along this axis. Along this axis, that regimen will provide a benefit to patients. And then along this axis is how those things, people's prognostic factors, whether they're going to do too well or too badly to benefit, play in to that. So it's a plane, and in certain areas that plane is going to go under, right? There's going to be negative benefit or more harm for some treatments. And this is a representation of biology, but what our clinical trial is trying to do is find the combination of these three major factors where you're actually going to be able to show a benefit that's meaningful. And that's what you're setting out to do. And that's pretty hard. But again, there is some underlying biology when we do a clinical trial, some biologic truth, and we never can fully explore what this surface looks like. But it is an underlying factor that, the treatments, unless the treatment is truly null and doesn't have any effect, then this thing is flat. There's something out there where if you get the right combinations of these things, you will find a important treatment effect. One very important example would be alteplase for acute ischemic stroke. One of the most important prognostic factors for a patient with acute stroke is the time to treatment, in addition to age and severity. So the NINDS-CTMC trial elected to motivate the scientific community in the United States by forcing half of the patients in the trial to be within 90 minutes of onset and the other half 91 to 180 minutes. So they were over here on this surface with a similar regimen. But in Europe, in the ECAS-2 trial, that degree of forcing was not, they didn't enforce that, so that maybe their patients were over here where there was less benefit because it took longer to open the vessels and patients were enrolled at a longer time course. So that they couldn't detect a positive treatment effect over there. So that is one example where, depending on where you are in this, you may have a drug that if you don't pick it right, if there wasn't an NINDS trial going on at the same time as the ECAS trial, it may have been a tough sell to look at further trials of TPA. But fortunately, there was, and we looked at this different space on this curve. So part two, trying to put this in the perspective of how, for drugs or other treatments, whether they're behavioral interventions or other things, they can progress through phases. There's this slide thinks about this from the drug perspective that, there's preclinical research working out target engagement mechanism. There's animal testing in the short term and long term, and some of that is toxicity if the compound hasn't been in animals before, but some of it's preliminary efficacy. For example, TPA was tested in a clot model of rabbits to help determine the dose and the time schedule. An IND is submitted to the FDA, and then there's the phase 1 clinical studies. We're looking at more pharmacodynamics, pharmacokinetics in healthy people, or if the compound is truly novel, or perhaps that patient population, if the compound has been looked at in healthy people, but is, as an MS treatment, but now is being looked at in stroke, there may be unique safety issues that would be different in the stroke population in some phase 1 studies, maybe people with the disease. And then phase 2 studies, which are a large bit of the exploratory space, potentially more solidifying dosing regimens and other major factors where a lot of the studies that are being done in this course are going to be. Then phase 3 studies, which are these pivotal studies that are determining if a treatment is efficacious. And then this new, if this is a novel compound, the new drug application is submitted and the FDA could approve the treatment. Again, sometimes we're repurposing agents that are approved drugs to learn about their efficacy in new places, and it may not be leading to a final FDA regulatory approval. It just would give us great guidance from a clinical practice perspective. For example, some of the studies looking at TNK as an alternative thrombolytic to TPA may not lead to a labeling change, or they might. But if they could change practice, even if they're not leading to a labeling change. A great example is the ECAS-3 study in stroke, which found efficacy between 3 and 4.5 hours. That was very practice changing, but it was not, at least to my knowledge yet, something that was used to change the label for alteplase in either North America or Europe. So again, thinking about these, this is sort of put a different way, like in terms of developing drugs, thinking of the pharmacology, thinking about how the therapeutic works in an exploratory space and then the confirmatory space. And then there's this idea of phase 4, post-marketing surveillance. One thing that's interesting is that safety issues with drugs and other interventions often are rarer than outcome events, which means that the smaller studies, the phase 2 or phase 1 studies, are going to be unlikely to show safety issues. For example, cardiovascular complications with particular glutazones for diabetes. So these phase 4 studies, the post-marketing surveillance time, can be a key way to look at those things. So in terms of part 3 of our talk, let me just pause here for any questions. Okay, I have a couple here. If the available preclinical data to support your interventions is not precisely aligned with your study design, how detrimental could that be to your proposal? So that's a really good question. I think you have to contextualize that within the confines and limitations of the research, and whether or not perhaps better aligned, you could convince collaborators with basic science labs to better align the research. I think it depends on whether there's a firm mechanistic basis for the treatment. And again, this might be, in drug studies, animal work. In behavioral stuff, this might be theory or observed behaviors or things other than animal studies. So I guess having things exactly aligned is a great strength, but having the overall idea, like for example, if I go back to the thrombolysis example, maybe you didn't range through the exact dose in humans in the animal study, but you looked at larger and larger clot burden to see how big a clot you could bust up, how much hemorrhagic transformation you were causing, depending on delay of treatment. So, if the concept is, that there is a time window and there is a dose that is most effective of breaking up big clots, I think that that's possibly a way of kind of getting at those. But I think you have to think carefully about it. And I think I will say though, on the other hand, in perhaps another setting, a study that looked at a neuroprotectant, it did okay at neuroprotecting rats for 3 days, it did better protecting rats for 5 days, and it did even better at protecting rats when given for 7 days. But when it moved to the human trial, it was like hard to give it for more than 3 days because they felt that people would be moving to acute rehab and other things. So they picked 3 days, but if I was reviewing that grant and I had access and the knowledge to know about the animal study that showed that, while 3 days was protective, 7 days was really protective, I would be critical of that grant. So I'd say, you don't need to completely honor the preclinical data, but think about the biology. And if you're ignoring something that could potentially be important for a reason that is, more logistical, and there might be alternatives, right? Could they transition to a sub-Q version of the molecule or an oral version of the molecule so you don't have to give an infusion for 7 days? You have to think about those sorts of things. And Dr. Gutman is eloquently pointing out it's very dependent on a variety of factors, including target engagement and what phase of study you're looking at. But and as you said, this is a major review criteria for an NIH clinical trials. And I think in order to do good science, particularly at NINDS, you really have to think through the underlying biological premise and have that firmly rooted in biology. So those are great questions. I will march along here. Whoops. So all right. When you're doing a clinical trial, you should have generally one main question. Is X better than Y? Or does X improve a patient-oriented outcome compared to Y when administered to this population for this long at this dose? Now, the question may be more couched towards feasibility. In the feasibility trial that was done prior to the randomized control trial that I'm doing on hypertension text messaging, we actually had a question of, if we hand out blood pressure cuffs in the emergency department and then text people for 3 weeks, how many people will text us back and how many will still be hypertensive? And our question and our sort of goalpost for that was, if more than a third of those people text us back and are still hypertensive, we are willing to hand out blood pressure cuffs and then enroll those who respond in a study to try to get their blood pressures down. So the main question in that study was, how many blood pressure cuffs do I have to hand out? What is the yield of handing out the blood pressure cuffs? Can you make the question better, right? What is the dose, schedule, or population that would hold the most promise for determining that treatment X can be proven to be better than treatment Y in a phase 3 trial of size N? So that's a pretty good question. Sometimes it can be hard to estimate this. But maybe we're asking too many questions at once here, right? That is a really good question, but I might need to have a 1,500 patient phase 2 trial, and there may not be a lot of enthusiasm for that. And it may be that you have to go back to animal data or other mechanistic data to try to reduce the uncertainty in these things. But, again, phase 2 trials are generally smaller. Maybe they should be larger. We are, I think, nudging in that direction, but it's not there yet. So you have to prioritize the questions that you're asking. And then what are the risks of getting this question wrong? FDA worries about declaring an ineffective drug efficacious. That's a big deal. That's a big deal, and that's their primary one of their primary issues. In academia, we need to worry about designing the study wrong and declaring something neutral when we were testing it in the wrong group and with the wrong parameters, population, dose, schedule. If we could have used more of the drug, would something different have turned out? And then, to put this in perspective, is what is an early phase trial? And generally, the concept is learning versus confirming. The learn space, phase 1 and 2, potentially greater flexibility in number of questions and goals, and potentially willing to tolerate higher error rates and sources of bias, but should not be set up as underpowered confirmatory trials. In the confirmatory space, phase 3, you have to have a pre-specified single primary question, a clinically meaningful difference in the outcome measure, which can be a little fuzzy at times, but still something that would motivate changes in clinical practice. And whether you say that's improving the rate of good outcome with TPA from 40 to 4% or from 40% to 44%, which is, I would say, given the number of people treated with TPA, would be a great advance. That type of effect size would require a very big study. So sometimes people are a little nervous about doing those sorts of studies. And this is often the one-to-one randomized trial. So just to quickly go through some of the questions that can be asked in terms of the early phase. These are some good questions for trials. For whatever reason, I put question 1 before this. Let me just look, see if we got other questions. Okay. I don't see anything else in the chat box yet, so we will go back to early phase question. Okay, can the drug be tolerated? And I would also say, can the intervention be tolerated, right? If this is and will it be done? Adherence is key for some non-pharmacologic interventions. If this is an exercise intervention, how much exercise are the people in your exercise group getting? This is a step intervention. How many people are getting 10,000 steps? What is the difference in the steps between your control and your intervention group? And if you're telling people they need to get 20,000 steps, how many people are hitting that target? But it can also be a dose escalation question. How much of the drug can be tolerated? And this differs greatly for people who are going to be on a chronic treatment versus a one-time acute treatment, because the types of problems are different for those different situations. For a thrombolytic, we talk about hemorrhage as sort of being a toxicity, and to some degree it is, but it exerts itself on the main outcome, which is, are they going to have a good clinical outcome afterwards? If they have a great clinical outcome and some petechial hemorrhage, I don't care. However, in say a ALS treatment that makes you throw up continuously all day, but it slows your ALS, if you manage to, let's say it's a shot, that might not be something that is tolerated at that dose because it causes them to throw up continually. There are some designs that have been used. The 3+3 design, we'll say for historical interest. These slides are from Roger. Continual reassessment method allows you to look at these things. These methods, though, are really looking for things that have pretty high levels of toxicity, like 20, 30%, to really kind of smooth that dose response curve to figure out when the bad thing from this drug happens, where is the point where it's happening at an acceptable rate. So another really good type of question for early phase is, can we establish proof of concept? Can we find that the drug's binding to the target receptor? Hopefully, if that's been established in animals, it maybe can be established in humans as well. And is it getting into the central nervous system? This could potentially be a dose-ranging study or a study of a biomarker or assay, perhaps some sort of imaging study that looks to see if the drug is getting into the brain. Another good early phase question is, what dose or doses should be investigated further? And here dose may be duration for some acute treatments, in addition to chronic treatments. Obviously, people are on a dose and maintained, but if somebody's had a stroke or traumatic brain injury, how long should you keep giving the drug to them to adequately get through the disease process you're treating? If you're trying to reduce apoptosis, do you really need to be giving that for a whole month, or is most of the apoptosis that's going to happen within sort of 1 to 7 days? And again, this is where it's crucial to go back to the biology and learn about those things. And then you're really trying to make this balance of efficacy and toxicity. And I think, and Roger and I put this on here, and I agree, sometimes the dose range is narrowed too early. And again, this could be the intensity of the intervention or device settings or schedule. We use the drug example as sort of the main idea, but questions in this space are relevant even for non-drug trials. The outcome that should be measured, and you can see this is similar to what we are talking about in our small groups. What is the effect on outcome, and is it something that's clinically important to patients? I think disability after stroke is clinically important, but some of the other gradations may miss other patient-centered outcomes. as well. Whether the outcome measures are validated and how reliable they are. But again, be careful if you're considering multiple outcomes. At least sometimes it's helpful to measure them, but the Dr. Thebane's lecture or webinar on pilot and feasibility studies really puts us out there in that being more observational on those, confidence interval estimation as opposed to doing a ton of hypotheses and getting yourself into multiplicity lands. Should you be testing multiple, the intervention and looking at what the ideal outcome is. In many diseases, though, the type of outcome measure is something that's well established. For example, the modified Rankin scale in stroke. And then what are the disease states that should be studied? And this is, this balance. If you look at too narrow a set of disease states, like I'm only going to look at stroke patients who have NIH stroke scales of 5 to 8, you're going to have fewer people you can recruit. And there may be other treatments and factors that they should be getting that can be problematic. But on the other hand, if the biological mechanism and the premise of your proposed treatment don't seem like they would work in a certain type of subject, then even if there's more of those subjects available, it may not be efficient to include them in your trial. And maybe in different, there may be multiple phase 2 trials of different agents or same agent in different diseases like subarachnoid hemorrhage, ischemic stroke, ICH. And one possibility is an indication finding trial. This is pretty innovative, where you can look at multiple diseases and use hierarchical modeling to bridge across those. Again, that's a very advanced way of looking at an early phase question. But if you've looked at some of the work in oncology where people have the same sort of mutation across different diseases, for those sorts of trials, that can be essential. But it's based in the biology. We know the tumors have the same protein that is being expressed through this mutation. So this can be done, and it can be potentially a good question if the biology is matched. Put back here in terms of the population. If you narrow the population too much, you have difficulties enrolling, but you may have less variability if you know what their natural history is going to be. If you have the broad population, you can have increased variability. And there might be heterogeneity of treatment effect. I think this is one of the things. There's concerns about how well TPA works in older adults, but when this has been looked at, there's not really a ton of heterogeneity of treatment effect. Older adults are at higher risk of major disability, but they do confer a benefit from thrombolysis or thrombectomy. So you want to inform the design of a confirmed phase trial, and you want to try to match your population in this early study. And if you have a hard time recruiting a specific subtype in your early phase trial, that might be knowledge that they may not be the best to include in the next step. One of the questions may be, how should a confirmatory study be designed, implemented, and analyzed? And again, you want to have your learn phase study conducted similarly, so you learn about a lot of the logistics, but you have to be asking a question that isn't an underpowered efficacy study in that space. Sometimes things known as futility designs can ensure that you're within a zone of promise, that your treatment does have an effect that would be potentially able to be detected in a larger scale confirmatory efficacy study of a certain size. And then your clinical trial, crucially, has to have a way of potentially giving you this output that is, maybe you shouldn't be doing the follow-up study. That is really important. If you design a trial that is going to give you, you're going to look at, 10 patients, 5 with TPA and 5 with a new thrombolytic, and see if maybe the hemorrhage rate's different. Since the hemorrhage rate is only 6%, guess what? The hemorrhage rate's not going to be different across those two trials, or if it is, it's probably random variation, even if they're exactly the same, unless the new drug has like 10 times the hemorrhage rate of TPA. So you are not going to be able to answer this question at the end of that study. Should I take this drug on? Should we move to a confirmatory study? It may be that that's something you need to do before you move on to a later exploratory phase study or learn phase study. But again, you have to understand what your decision point is going to be, and you may be able to predict the probability of a positive confirmatory study depending on the remaining uncertainty. Now, the counterexample is we've looked at many trials to prevent Alzheimer's disease, and some of them have had some good looking probability of positivity in phase 3, and they haven't turned out that way. All right, so I've got 3 minutes. Let's see, did we accumulate any questions? All right, no questions. I should be able to wrap up quickly, and then you can start typing questions in now. One thing that I would like you to consider doing with your infinite free time is if there is a type of design you're thinking of, whether it's a pilot and feasibility trial, for that I would definitely look at the CONSORT guidance extension on pilot and feasibility trials. If you're looking at a non-pharmacologic intervention, I would look at the CONSORT extension for those. And again, this gives you a lot of the ideas and things that you should be thinking about in your design and can be very helpful. If you're thinking of a cluster trial or a set of N of 1 trials, this is a good place to maybe do a little preliminary reading. So I would say try to check out the CONSORT extensions for the type of design that you're going to be doing. And you could always look at regular CONSORT, although that is a bit couched towards 2-arm confirmatory studies, which most of you are not going to be designing within this course. So, again, the default maybe is the pilot and feasibility trials one to kind of look at that. But again, that is something that I think you should check out. So quickly, in conclusion, early phase clinical trials are hard. Getting the question right is crucial. And it can be challenging to prioritize these questions. But again, it's important for you and for the scientific community to be thoughtful about how we design these studies. With that, I will open up the chat box. And I think if there are other questions, yes, Dr. Ungarun has asked if there is a good reading resource relevant to this talk. We will send something out. I can think of some things. I know Roger Lewis has some that he likes about that. We will. I can't come up. Yeah, well, I think that's Dr. Conway brings up a great point. In the syllabus, one of the early assignments is to look at the NINDS and NIH rigor criteria, looking at how you evaluate preclinical evidence and also how you do transparent and reproducible clinical research. That is a good thing to read. I guess the other, even, some of you may not be doing quite pilot and feasibility trials, but I do think if you search on Google Scholar for pilot and feasibility trials, the article by Dr. Thabane that was cited about 1,000 times is really a good one to look at because it has many of the pitfalls. And this is, depending on where you were, at what phase you are with your research, maybe you've done a pilot study, but then if you read this article, you'll be like, oh, there were some other things I should have looked into that, maybe in your next phase study, you can kind of address some of those questions. Or if you're designing something that would be more in the pilot and feasibility space, it gives you a lot of good pointers for how to orient your hypotheses, outcomes, and the sorts of questions you should be asking. All right, so I have 1 o'clock, and Joy, I guess we will have to send them a link to the webinar evaluations after we get that up on Qualtrics.
Yeah, okay.
I think we have this. We should have this one listed, and if not, we'll make sure it's on the list so that you can select it. Again, looking forward to everybody starting their small groups, and please, reach out to us if you have questions. And again, thanks for your attention today, and looking forward to having you all in the course.